Today I'm going to introduce my past and current work on the shell-shape evolution in Galapagos tortoises. So why to focus on the shell of the tortoises, of the turtle in general? The shell is a very important fitness-related trait for this animal. It offers protection, help maintain optimum homeostasis of the metabolites. It's correlated with optimal locomotor performances and optimal mating, among other things. So the work that I'm going to present today, and which I continue to work on, is done mostly in collaboration with 4 people. One is Julien Claude at the University of Montpellier in France, Adalgisa Caccone at the University of Yale in the US, Benjamin Gilles at the LIRM in Montpellier, France, and Ari van der Meijden here at the CBO. So when we talk about distinct morphologies of the Galapagos tortoises, we mostly refer to 2 different shell shapes. The first one is the saddleback that you see here on the left. The animal have this characteristic very high anterior opening of the carapace. So the neck can extend very high. The tortoises are generally also smaller inside, and they are slender on the sides. Then there is the shape on the right, which is a classical dome tortoises with this cupola-like carapace. And then there is a third type, the aplastrado is called, which was once very common on the island of Isabela, which is the largest island of the Galapagos archipelago. But unfortunately now there are only a few individuals remaining with this morphology. So in my talk I focus on the 2 main different shell shapes, the saddleback and the dome. So these different shell morphology occur on the different islands of the archipelago. As you can see on the right, these are the island where the Galapagos tortoises occur or were before getting extinct. And the 2 different symbols represent the 2 different morphology, and this different morphology occur on many different islands. And if you plot this distinct shell morphology on a mitochondrial phylogeny, I don't expect you to read this phylogeny, but just look for the different, this drawing, the shell evolved multiple times. So I have to be precise and tell you that saddleback morphology occurs only in the Galapagos Island. You don't find it anywhere else, although somebody claims that it's in the Aldabra atoll, but not really. So when you look at adult tortoises, the adults have these 2 different morphologies, saddleback or dome. But when you look at the juveniles look very much alike, and they have this very compressed dome shell shape. These are 2 different islands. These are the adults from the same island, and these are the juvenile. So when you look at the juvenile, at least for me, it's almost impossible to distinguish them in what they will become without having previous information. In the Galapagos Island, there are individuals that are raised in captivity, and what we observe is that if you take the juveniles of one of these 2 islands and raise them under the same environmental condition, they still turn out to be either saddleback or dome. And this would suggest that plasticity is limited, if not existing, in shell shape in Galapagos Island. So one of the things on which I'm currently working on in collaboration with Alison Llerena at the Charles Darwin Station in the Galapagos in Ecuador is to study the development and the shell growth to see if we can actually pinpoint during the growth a difference at which moment the 2 shells start to diverge. So we have eggs that are taken from the wild and raised in captivity. We have hatchling, and the individuals are measured every 4 months. And we have data from the 60s for many lineages of saddleback and dome tortoises. So, but when we are interested in studying phenotypic variation, so for example, a variation in the shell shape of the Galapagos tortoises, generally speaking, the factors that can contribute to this variation are either a different genotype, so different genes, or an environmental influence. So what we did, the first study that we did was to analyze how genetic variation in 2 lineages of Galapagos tortoises was paralleling or not the phenotypic variation in shell shape. What we did was to go on the island of Santa Cruz and analyze 2 different lineages of both dome Galapagos tortoises. So those lineages are on evolutionary tree recovered very in two different, very distinct clades. One is here and one is there. And because the taxonomy of the Galapagos tortoises is not clear, I just refer to them as different lineages, and I call them with the population name, Sierra Fatale and La Reserva. So these two dome tortoises occur in similar habitat, but they differ very much in population size. So Sierra Fatale is a bottleneck population and is estimated to contain about 100 individuals, while La Riserva is considered one of the largest population of the island with about 2,000, 3,000 individuals. And they also differ very much in the distribution area, with La Riserva having a very large distribution area. So what we did was to collect mitochondrial data, microsatellites data, and morphological data, as you can see here, for the same individuals. And the morphological data were collected on the carapace, so the top part, and on the plastron, which is the ventral part of the shell. And what we observe is that the population that has reduced genetic variation, Sierra Fatale, also show reduce size variation and reduce shape variation. So in general, what we can say is that there is a parallel between the intra-lineage genetic variation and the shell shape variation and size variation. But this parallel between the ratio of the genetic variation and the ratio of the phenotypic variation between the two lineages is not of the same level. So this would suggest that because there is much less phenotypic variation, maybe there is a constraint on this variation. But to better understand the direction of variation, we decide to look at how this variation is organized in space. So because these shells are three-dimensional structure, we develop three-dimensional capture method that we applied to the same population. And so what you see here is the mean shape for each of the two lineages. The gray one is La Reserva and the black one is the other, is Sierra Fatale. And you can see how the variation, the direction of variation between the mean shape. And one of the things that we observe is that La Riserva, the gray one, this one, has a posterior that is slightly higher than the other one. So now, because there's been data on tortoises relating difference in shape with different level of migration, and because La Reserva has a larger distribution area, we speculate that this higher back part of the carapace may facilitate movement of the legs, and so may be connected with higher dispersal pattern. But we don't have data for this. There are studies on movement of these two lineages going on right now in the Galapagos. So when you hear talking about what are the causes of different shell morphology in Galapagos tortoises, almost everywhere, absolutely everywhere, you read what the saddleback and the dome tortoises are adapted for different environment. So the hypothesis is that saddleback tortoises do better in dry environment, while dome tortoises do better in wet environment. And this is due to the fact that there is effectively a correlation between where the tortoises, the two morphotypes, mostly occur. You find mostly saddleback tortoises in the drier part of the island and dome tortoises in the wetter part of the island. But this has never been actually tested and demonstrated. As other hypotheses that there is a difference in performance for biomechanical factor or competitive behavior, and also has also never been tested. I'm actually going to come back to this last one and explain it better in the last part of the talk. Of course, all these factors say that the major evolutionary force shaping shell difference in Galapagos tortoises is selection. But the competing hypothesis is that actually the difference is due to just neutral evolution, so genetic drift. So to test for this hypothesis, so to test that the major evolutionary force associated with this difference in shell morphology is actually neutral evolution and genetic drift, we are going to use the matrix of variance, covariance of phenotypic traits. And we compare these matrices of variance, covariance between and among groups with different morphology. So I collected a very large data set of 3D data for all the extinct and extant lineages of Galapagos tortoises. And I'm going to apply these methods. I'm currently working on it, so I cannot show you with some results. But independently of the mechanism, of the main evolutionary force acting on the shell shape difference in Galapagos tortoises, I'm also interested in testing if it's true that the two different morphotypes are associated with different performance for a function. And so the first things that we decided to test was if there is a different performance in self-writing capacity. So what is a self-writing? Self-writing is this. Basically, tortoises walk all the time on this lava field, and they fall on their back. It happens. So when they fall on their back, it's very important for them to turn very quickly on their 4 feet, as they could die due to lung compression, and especially because of the heat. So the theory is that saddleback tortoises turn themselves much quicker because they do live in drier environment than the dome tortoises. And we are actually interested in testing if this is true. So there's been a study published recently in the Proceedings of the Royal Society, and just using mathematical model, they model different shell shape for different species of turtles. So this is all due to mathematical model, and what they observe is that depending on the shell morphology, the technique that the turtle use for self-writing is different. So some only rely on the shell morphology. Some other also has to use bobbing of the head and the legs to actually move quicker. So what we plan to do is to use this 3D reconstruction and actual data to test if in the Galapagos tortoises we have this difference in self-writing. One important thing that they did in this study is they assume that the center of gravity is in the center of the shell. This is an assumption which may not be true. And actually, where is the center of gravity is really important to establish the correct self-writing. Because, of course, if the center of gravity is here or here, it changes your movement to return to your stability. So the first thing we did was to establish where is the center of mass in a Galapagos tortoises. And here you see the setting that we used. Basically, what we did was to build 2 platforms like this, which are separated by load cells. Load cells are like scales that measure the weight. And there is also a jack to lift the upper plate like here. So estimating the center of mass in a 3D space in a 2D system is fairly easily. You can do it on a plane with this 3 scale. But to also measure the 3rd dimension, the height, you need to lift it so that the center of mass move, and you basically can calculate the 3rd dimension. So and here you can see the actual setting. So we were at the Rotterdam Zoo in the Netherlands. This is the bottom plate with the load cells, the jack. Here is Dr. van der Meyden, who is actually testing the stability of the 2 plates. And of course, when he was on the plates, everything was fine. But when you work with animals, they don't necessarily do what you want them to do. So as soon as we try to get the tortoises on the plate, the 150 kilos of the tortoises went all on one load cells. And this caused a panic of 10 seconds because we thought that they crushed the load cells. So the next step was actually lifting the 150 kilos directly on the plate. So what we found is that the center of mass is actually not in the center of the shell as has been previously thought. So these 4 points that you see here are actually the top platform. This is get up to here. These points here. So basically, you have the plastron attachment here, and as you can see, the center of mass is much closer to the plastron than to the center. And this is probably because, of course, you have organs that are not uniformly distributed into the shell. So now that we know where the center of mass here is, what we are going to do is, with the 3D data that we have for dome and saddleback species, we are going to simulate applying forces and pulling the different shells until they turn back. And then we basically statistically test which stronger forces you need to pull, if the dome morphology or the saddleback, so that we can estimate if there is actually a difference in performance in self-writing connected with the different morphology. So, and to conclude, I would like to thank all the people who helped us at the Rotterdam Zoo for the calculation of the center of mass and the funding societies. Thank you very much.
Thank you.